In today's video, we are going to be considering chromosomal mutations. Remember, we talked about that there are different types of mutation. We have the frame shift, the point mutation, the chromosomal mutation. So basically talking about the chromosomal mutation, and there are types today. And there are two basic types of chromosomal mutation. They are the aneuploidy and the polyploidy. Alright, now basically chromosomal mutation is usually with respect to two things. It's either that the number of chromosomes change, which is what we have been talking about here in aneuploidy and polyploidy, or there's a change in the shape and size and the rest of that about the chromosome. So basically here we are talking about the change in the number of chromosomes, alright, due to some conditions. Alright, now we are going to basically be talking about aneuploidy first. Aneuploidy is gotten from two Greek words, which are aneu and ploidy. So you remember ploidy basically talks about the number of chromosome sets, ploidy, number of chromosome sets that an individual has, while aneu means abnormal. So to combine both, it means that abnormal set of chromosomes. While polyploidy means many sets of chromosomes. Now what happens in aneuploidy? I'm going to use these diagrams to explain what happens in aneuploidy. You already know that a diploid organism has two sets of chromosomes. So for human we have 23 sets, which amounts to 46 total in total of number of chromosomes. So it means that our haploid number should be 23. Our haploid number N should be 43. And our diploid number 2N is 46. Okay? So, and for fish such as tilapia is 44. But I'm going to be using human for the sake of this tutorial. So, now I've chosen out of the 46 sets of 46 chromosomes, I've chosen only 4 sets of chromosomes. That is, I'm working out of 46, I'm working with only 8 chromosomes here, 8 sister chromosomes here. Alright, so now what happens in meiotic M1 anaphase, that is anaphase 1, and this is anaphase 2. Now what happens in M1 anaphase, that is anaphase 1, that is meiotic division anaphase 1. Now aneuploidy can happen with both allosomes and autosomes, but here I've chosen allosomes because we will be talking about fertilization as well. So the individual started with 8 chromosomes, so meaning that our N, as per this example, our haploid number should be 4 as per this example. Our haploid number should be 4. So naturally at anaphase, having assembled at metaphase here, at anaphase, these chromosomes, sister chromosomes should separate. Non-sister chromosomes here should separate. Alright, you know why it is non-sister chromosome? Because of exchange recombination might have occurred. So they should separate. But look at the first one. The first set separated equally. The second set are separated equally. The third set separated equally. But look at this one. Because of the absence of this spindle fiber, that is a condition known as spindle assembly dysfunction. When there is no spindle fiber formed here, it causes a non-disjunction should occur. So naturally, they should disjoint. They should be disjointed here because they will move away. But here, there will be no disjunction to happen at this last set. All right. So what would that result into? We have two sets of two daughter cells being created, of course, in meiosis 1. Now, you will now notice that the daughter cell that is formed, one of the daughter cells is going to have an extra copy of a chromosome. So look at this, it has an extra copy. So you have one, instead of having an N number of 4, here you are going to see our N here is supposed to be 4, so we have 1, 2, 3, 4, 5. So it means that this person is having 4 chromosome sets here at the end of meiotic 1 and 3 here. This one has is deficient of one. All right. So when the further division takes place, then the sister chromosomes that will now have will be one chromatids rather. Now we now have 1, 2, 3, 4, 5. So we now have 5 chromosomes, which n number is supposed to be 4 actually. So you now notice that this one is 5, meaning that 1 has been added. One set of chromosomes has been added. I'm going to talk about what this means. It's called hyperploidy and aneuploidy. It's a type of aneuploidy where our haploid number gets increased. While in polyploidy here, in this second, so there are 2 sister cells that are n plus 1 and n plus 1. Alright? And here we are going to produce, instead of having the n equals 4, we have 1, 2, 3 cells. So that will be n minus 1 and n minus 1. Alright, so that is what happens with aneuploidy. Now, there is now a possibility of syngamy with different chromosomes. Now, if this guy, so let's say this particular guy fertilizes another chromosome, another cell, haploid cell that is normal. If it's fertilizer a normal haploid cell, it's going to produce a normal haploid cell should be n, right? So n plus this n plus 1 becomes 2n plus 1. So the individual becomes 2n plus 1. And this is a condition known as trisomy. This is a condition known as trisomy. While if this one fertilizes a normal cell, if this one fertilizes a normal n cell, it's going to be n plus n minus 1. And that will be 2n minus 1. This is a condition known as monosomy. Alright? So, but if it fertilizes such one as this, that experiences this kind of division, is going to be n minus 1 plus n minus 1. And that will be 2n minus 2. That is called nullisomy. Alright? If it fertilizes this sort, it's going to produce a normal chromosome, a normal zygote. It's going to produce a normal zygote. While this one produces 2n plus 1 zygote, this one produces 2n minus 1 zygote. Alright, now let's quickly get to polyploidy because I'm going to explain some other things about aneuploidy. Now, polyploidy is, now you see what is that? There is no spindle fiber at all formed here. So no disjunction in meiotic division 1 here, as happening here, M1 here. There is no disjunction at all. Alright, now this doesn't mean that the non-disjunction cannot take place in M2. Non-disjunction can also take place in M2. It is not really in M1, but I decide to use M1 as where the non-disjunction is taking place. Alright, this process is spontaneous. So if M1 takes place here, if non-disjunction takes place in M1, then it's going to produce 2 daughter cells with one not having any chromosome at all. No chromosome at all. Which will also in turn produce daughter cells with no chromosomes at all. Now, we are not particular about this. The whole set of chromosomes is here. So meaning that this guy is still having 2n here. So when they now get disjointed here, in M2 phase, when they get disjointed in M2, then what do? They produce a chromosome that still has the 2 sets of chromosomes. So instead of producing an haploid, they produce a diploid chromosome. Alright, that is in polyploidy. So you see, instead of producing an N, which is an haplontic cell, it produces a diplontic cell. Such that if this diplontic cell now mates with a normal cell, a normally divided cell, which should be haplontic, then it produces what? A 3N. This is what we call triploidy. Triploidy. So a triploid is produced. While if it mates with such one as itself, it produces a tetraploid. Tetraploid. And this would continue to propagate. Now it should be noted that in some organisms such as humans, triploid or tetraploid do not exist. The hybrid is non-viable, will not survive. The zygote formed will not survive. But it's common in plants. It's common in glands. All right. So we already know the types of and we've known what happens in aneuploidy and polyploidy. Now let's now talk about the types. The types. So for example, in aneuploidy, there are two types. We have the hypoploidy and the hyperploidy. In aneuploidy, we have the hypoploidy and the hyperploidy. Now, what happens with hyperploidy and hyperploidy is this. There is a reduction in the normal number of chromosome sets. So what you are going to have is a 2n minus 1 at the end of the day. Or in the actual gametic cell, we are going to have n minus 1. So this is hypoploidy. And there are two types of hypoploidy. We have the monosomy, which is 2n minus 1. For the zygote formed, and the second one is nullisomy. Nullisomy is basically 2n minus 2. That is for the zygote formed. All right. Now we also have in humans, a typical example of this monosomy is when there is a deletion. Example is the Cri-du-Chat syndrome. The Cri-du-Chat syndrome, where there is a deletion of. You remember when we discussed chromosomes, we talked about the P-arm and the Q-arm. So when there is a deletion of this P-arm for a chromatid, let's use a chromatid or a single strand of chromosome. When there is a deletion of this P-arm, then it's a result of the 5th chromosome of human. The syndrome is called the Cri-du-Chat syndrome. All right. Another type of syndrome that exists in human is the Turner's syndrome. Turner's syndrome. Remember that we talked about the fact that it is the male sex is determined by the male chromosome in human. So, and that is because of heterogamity. So heterogamity exists in the male. So meaning that an absence of, and it is this Y chromosome that tells us it's a male. So meaning that an absence of the Y chromosome will mean that the organism will definitely be a female. So a female that doesn't have, and a female naturally in the human should have XX chromosome. Now if this other X chromosome doesn't exist, whereby the individual has X0 chromosome, then it's called Turner's syndrome. So notice one chromosome is lost, 2n minus 1. Alright. Now in hyperploidy is a condition where there is an increase in the number of chromosomes. There is an increase in the number of chromosomes. So, and that is what we talked about in trisomy and rare. So there are examples, of course. So there are examples. Now, of course, you should know that this. So when we talk about the Cri-du-Chat is just about we can classify hypoploidy and hyperploidy into two as well. The one that is autosomal and the one that is allosomal. This one is autosomal, while this one is allosomal, meaning sex is dealing with the sex chromosome. Alright. So for hyperploidy, we have the trisomy and the tetrasomy. Now this has been seen in humans. Alright. So in the trisomy is basically there is an addition, hyperploidy. We said an increase. Hyper means high, then hypo means low. Alright. So there's an increase in the number of chromosomes by 1. That's why it is trisomy. Then here there's an increase in the number of chromosomes by 2. Alright, typical example in human is what we call the Down syndrome or trisomy 21. The Down syndrome or trisomy 21, which is basically an addition. There is an extra chromosome in the chromosome 21, such that instead of having 2 chromosomes in chromosome 21, there is one other extra chromosome. Alright, so that is the Down syndrome. There is also the Edward syndrome. There is also the Edward syndrome. Alright, and there is also the Patau syndrome. The Patau syndrome. Basically, this one is on chromosome 21. This one happens on chromosome 18. Why this one happens on chromosome 13 or there by here? The Patau syndrome. It happens on chromosome 13. Now, there is also the tetrasomy. The tetrasomy is basically an autosome. When it happens with the autosome, it produces an extra pair of chromosomes, such as we see in the Klinefelter's syndrome. In Klinefelter's syndrome, what we notice is that there is an extra pair of it's called the Klinefelter is called the super female. So such that we have the XXX chromosome. Now you see there is an extra duplicate of in trisomy you are going to see in the autosome that is in the allosome. What happens is, if it's allosomal, what happens is Klinefelter's syndrome. But in tetrasomy, what happens is extreme Klinefelter, not just Klinefelter. Here, Klinefelter occurs on the allosome, so you have XXX superfamily. While in the extreme case, you are going to have 4 X's. Alright, so that is that. And there's also the super male. The super male, in the allosomal case here, the super male is going to be X, Y. The super male is called the Jordan syndrome. So this one is called Klinefelter syndrome. This one is called Jordan's syndrome. You're going to have X, then Y, where there's an extra pair of the Y chromosome. But when there is even an extra pair, so such that it's called the extreme super male. So in the extreme super male, you're going to have X, Y. Alright, so that is the extreme super male. Okay, so that's basically what happens with aneuploidy. Alright, how about polyploidy? In polyploids, there are 2 types of polyploidy. We have the autopolyploidy. And the allopolyploidy. Now, if the polyploidy, if the polyploidy happens naturally, it's going to be called allopolyploidy, that is naturally. But if it is a process of, if it happens as a process of breeding, through a process of breeding, that is by our artificial crossbreeding, it will be called allopolyploidy. And take note that since it's happening naturally, it's going to be within the same species. Why in allopolyploidy? It could be the interspecific. So this one is going to be intraspecific. This is going to be intraspecific. While this could be interspecific as a result of our human, breeding, crossbreeding. Alright, so that's basically it for chromosomal mutation. Please do well to subscribe to the channel. It helps the channel a lot. I keep on posting stuff that will help you understand biology and fisheries and do well in all areas of genetics. Please do well to subscribe to the channel. God bless you. I thank you.